Good evening, everybody, and welcome all of you to this live program at Orthopaedic Principles. Today, our guest of honor is Dr. Rishi Dhir from Harlow, UK. Dr. Dhir is a consultant upper limb surgeon at the Princess Alexandra Hospital in Harlow, United Kingdom. After completing his training in the Royal London Rotations, Dr. Dhir completed fellowships in Melbourne, Australia, and the Wrightington Upper Limb Unit in Wrightington, United Kingdom. He's also the CEO of Let's Talk Doctor, a unique course that trains FRCS candidates throughout the world. Most of the candidates who have been part of the course have cleared the exam and also secured high ranking. If you've noticed, Dr. Dhir has delivered a couple of lectures on our channel and it's already reached a huge audience. And today it's my great honor to bring back Dr. Rishi Dhir for this wonderful live program. Over to you, Rishi.
Thank you very much, Professor Gopalan, and thank you to everyone in India who's watching and listening. So what I thought I'd talk to you guys about today is an area which I think is often sort of neglected or not much attention is paid to it, and that's genetics, embryology, and the fisus. Now, for the basic science component of the exam, you don't need to know it in a lot of detail, but you just need to have a basic understanding and how to apply it to the clinical scenarios that you might meet. So let's start with genetics. And the contents of the talk, I'm going to talk to you about the basic structure and function of genes and cells. I'll talk to you about some of the abnormalities when things go wrong, and also modes of inheritance. So starting with the structure and function, we look at the chromosome. And the chromosomes are structures found in the nucleus which contain genetic material. So that's DNA, RNA, histone, and non-histone proteins. Now, the karyotype, which is the makeup of a cell, has 46 chromosomes in a normal somatic or body cell. And that's made of 22 pairs of autosomes, which are your body chromosomes, and one pair of sex chromosomes, which can be double X in females, XY in males. And if you look at a microscopic level, a chromosome consists of a short P arm and a longer Q arm, which is separated by a small constriction called the centromere. So guys, just remember that sort of basic structure before we move on. And the location of this centromere basically subdivides chromosomes into four basic shapes that you meet. So metacentric means it's roughly in the middle. So the P arm and Q arms are roughly the same length. Submetacentric means that the P and Q arms are of different lengths. Acrometacentric means that the centromere is virtually at one end. So you have very small P's and much longer Q arms. And the telocentric is where it is completely at one end. So there's no P arm and only a Q arm. We then move on to DNA, and let's look at the components in a bit more detail. So DNA stands for deoxyribonucleic acid. And basically, DNA sequences are made of three bits. So DNA is made up of a nitrogen base, a phosphate backbone, and sugars. And these DNA sequences are arranged in a specific order to form genes. So the way that I think of DNA is it's a polymer of deoxyribonucleotides composed of a nitrogen base, sugar and phosphate backbone. And these bases contain genetic information which are derived from purines and pyridines. The purine bases are adenine and guanine, and the pyrimidine bases are cytosine and thymine. And it's important to note that DNA has a double helix structure with paired bases in the center joined by hydrogen bonds. So double helix is the buzzword I want you guys to remember. And certain bases can only pair with each other. So adenine can only pair with thymine and guanine can only pair with cytosine. So the next thing is, how do cells grow and divide? And the buzzword to remember is mitosis, okay? So in mitosis, let's say you start with a mother cell. The first thing that happens is DNA will replicate by unwinding the double helix, allowing a complementary daughter strand to be formed using the original as a template. And this is called semi-conservative because each new helix that's formed contains one of the original chromosome strands. So every single chromosome replicates, moves to the center of the cell, and then splits apart. So you get these identical chromatids, and these are found in each daughter cell. Okay, so remember that replication moves to the center of the cell, splits, and then moves apart to form identical daughter cells. Okay, it's semi-conservative replication. We then move on to RNA. Okay, and the important point is, how do we convert genetic material? So the DNA gives us the genetic material, but obviously it's the proteins which carry out the function. So how do we convert this genetic material into proteins that translate into biological function? So there's two words you need to know: transcription and translation. So transcription is where you create templates from the DNA in the form of messenger RNA, mRNA. And it's done by an enzyme called RNA polymerase. The next thing that happens is the mRNA will exit the nucleus into the cytoplasm and combine with other RNA molecules called ribosomal RNA and transfer RNA, which allows protein synthesis to occur at the ribosomes. And that process is known as translation. So remember, transcription is the production of the mRNA, and that's conversion of the DNA into mRNA, and then the mRNA moves into the cytoplasm and combines with ribosomal and transfer RNA to form a protein. There's a couple of other words I want you guys to remember. So replication is where you have DNA being converted to DNA under the enzyme DNA polymerase. Transcription, as I've mentioned, is DNA being converted to RNA by the enzyme RNA polymerase. And translation is where the RNA is converted to protein. So we described our basic structure and function, what happens normally. The next thing is what happens when things go wrong. So chromosome abnormalities, you can think of them as either the level of the whole chromosome called aneuploidy or structural abnormalities of a single chromosome. So whole can involve loss or gain of a whole chromosome, which is called aneuploidy. And usually the loss of a whole chromosome is more severe in phenotype than a single gene defect, as you lose more genetic information, okay? And there's two types that you see. Monosomy is where you lose a chromosome, and it's only survivable if you lose an X chromosome rather than if you lose one of the body chromosomes, the autosomes. So for example, an example of that would be X0, called Turner's syndrome. And that's just some of the features that you typically get in Turner's syndrome. So for example, webbing of the neck, low posterior hairline, a heart-shaped face, typically short structure, coarctation of the aorta. You get this cubitus valgus deformity, a broad-based chest with widely spaced nipples, and they typically have amenorrhea and infertility. Okay, so that's Turner's syndrome, XO. The other whole chromosome abnormality is called trisomy, and that's where you gain a chromosome. And the common example we hear about is Down syndrome, trisomy 21. So typically you get features of mental retardation, characteristic eyes, congenital heart disease, intestinal defects, wide gap between the first and second toes, or you can have Klinefelter disease, which is basically where you gain an extra X chromosome, so XXY. Okay, so it's seen in males, but they'll typically have female characteristics. So lack of body hair, gynecomastia, female-like hips. They get lack of pubic hair, testicular atrophy, or infertility. So these are the typical features in Klinefelter syndrome. So those are your whole chromosome abnormalities. We next move on to our structural abnormalities. And these are where you have the changes in the chromosome structure involving one or both copies of a gene. And there's some important phrases that you guys need to know for your exam. So the genotype is the inherited genetic code of an individual, whereas the phenotype is the clinical manifestation of the genetic code. So how do they actually present clinically? Disorders which occur at specific alleles disorders occur, at specific alleles at one or both autosomes. And an allele is defined as one or two alternative forms of a gene known as mutations that appear at the same locus or spot on the chromosome, and they control the same characteristics. So for example, you may have alleles for blood type. When both alleles are similarly involved, we call that homozygous trait. If they differ, it's called a heterozygous trait. And a mutation can occur at specific nucleotide bases. So you can get point mutations, or larger parts of the chromosome can be involved, called deletions, where part of the chromosome is removed. Inversions are where part of the chromosome is basically rotating about 180 degrees, or translocation is where genetic material is transferred from one chromosome to another. So remember those words: point mutation, inversion, twisting 180 degrees, or translocation. So we've described about basic structure and function of chromosomes, abnormalities that you might meet in the exam, and let's move on to modes of inheritance, okay? Inheritance is quite important because this is really where we come in as doctors. So meiosis is the buzzword I want you guys to remember. And meiosis is a special type of cell division which only occurs in the sexually reproducing organisms to produce gametes, egg or sperm cells, okay? It's got 2 rounds of cell division, so that's a difference from mitosis, and that ultimately results in 4 cells with a single copy, a haploid, of each chromosome, 1 copy of each autosome, and a single sex chromosome. Now, one thing I want you guys to remember is, prior to the division happening, genetic material from the paternal and maternal copies of each chromosome crosses over this thing called the chiasma, okay? And that creates new combinations of a code on each chromosome. So you can see here, there's several different combinations, okay? Because of this crossing over of genetic material, okay? And a child will inherit 1/2 of their chromosomal pair and 1 sex chromosome from each parent. The mother can only contribute an X chromosome, but the father can contribute an X or a Y chromosome. So before meiosis begins during the S phase of the cell cycle, the DNA of each chromosome is replicated. So that's the first stage. So it consists of 2 identical sister chromatids which remain held together. We call that the pre-meiotic or S phase. So remember, step 1, genetic replication or DNA replication. And immediately after DNA replication, the meiotic cells enter a sort of prolonged G2-like stage. We call that the meiotic prophase. And during this period here, 2 things happen. So the first one is you get crossing over of alleles between these homologous chromosomes, and that allows exchange of genetic material. And the place, as I mentioned, where the crossing over occurs is called the chiasma or chiasmata, plural. So that's the first word, crossover. The second thing that happens is you get independent segregation. So you get random pairing up of these homologous chromosomes. It can occur in several different ways, okay? Then in meiosis 1, the chromosomes are pulled apart, these homologous chromosomes. In meiosis 2, the chromatids are pulled apart, okay? So DNA replication, then pairing, then exchange occurs. So each chromatid will be different, as you can see here. Some have blue, some have purple, okay? So that's the important point, all right? So that means 4 chromatids are put into the gametes, and the prophase is where you get the crossing over. So remember those 2 words, crossing over and independent segregation. Then in meiosis 1, the homologous chromosomes are pulled apart. Each homologous pair is separated. And then the second phase, meiosis 2, is just like mitosis, okay? So you'll end up with 23, basically 22 homologous autosomes, 22 autosomal chromosomes and 1 pair of sex chromosomes, okay? So those 2 things, independent segregation and crossing over, are your sources of genetic variation. And that's the thing I want you to remember for the exam. So what about defects? What happens when things go wrong? Well, in terms of inherited defects, it can be a single gene that goes wrong, or it can be polygenic inheritance, okay? And a single gene is called Mendelian, and polygenic is non-Mendelian, okay? And the example of that may be Autosomal Dominant, Autosomal Recessive, Xlinked Dominant, and Xlinked Recessive. Those are all forms of single gene inherited defects, okay? Now, guys, remember, Mendelian is key for the exam, okay? Those 4 types, you need to be able to draw tables of inheritance or Punnett squares for each one. So it's very important to learn these. So basically, you need one abnormal gene from each parent to inherit the disease in autosomal dominant. That's the first one to remember. So autosomal dominant basically means if you get one abnormal gene from one of the parents, you're going to have the disease, all right? So the question I ask you guys is, if the father is affected in this case and the mother is normal, what percentage of children will be affected in this autosomal dominant, okay? So let's imagine that the father basically has let's say the disease gene is a big H, okay? And he has big H, little h, and the mother has little h, little h. So she's normal, okay? So what will happen is one of the children will have big H, little h, and be affected because they'll get the big H from the dad. The other one will be big H, little h, because they'll get the big H from the dad. Whereas these 2 children will get the little h from both parents and will not have the disease. So in this case, 50% of the children will be affected, okay? Remember, with autosomal dominant conditions, cases are usually heterozygous. You get one copy of the diseased gene because homozygous is usually fatal. And examples of this would be Marfan's, achondroplasia, neurofibromatosis, type 1, osteopetrosis, the tardiforms, any of the genetically inherited diseases, the majority of those that we meet, the dysplasias, are type 1. That's the thing to remember, okay? So the key thing is if one parent has a condition, the probability of inheritance is 50%, and the ratio of male to females affected is 1 to 1. Now, autosomal recessive is slightly different. In autosomal recessive, you need 2 copies of the abnormal gene to inherit the disease. If you have one, you're known as a carrier, for example, sickle cell disease. And if both, so the question they might ask you is, if both parents are carriers, what percentage have the disease and what percentage are carriers, okay? So in this case here, let's say big H, little h, they've got the recessive gene, but they don't have the phenotype. So they're both carriers. In this case, we can see here, so if we think of the black is the normal gene, the blue is the abnormal gene. This boy here gets the black from the dad, black from the mum, so it's unaffected. The daughter gets the blue from the dad, black from the mum, so she's a carrier. The son gets the blue from the mum, black from the dad, so it's a carrier, and the daughter gets the blue from both, so affected daughter, okay? So in this case, if both parents are carriers, 50% of the children will be carriers, and 25% will have the disease. The ratio male to female is 1 to 1. So you will need to be able to draw these out, guys, these Punnett squares for each of the conditions. Now let's move to X-linked recessive. And these occur more commonly in males, and that's because males only have one X chromosome, and therefore a single recessive allele on the X chromosome cannot be masked by the Y chromosome in females. So basically, in this case, homozygous are affected, heterozygous are carriers, okay? Females will usually be carriers because they'll have a normal allele on one X chromosome and an abnormal allele on the other. If females had abnormal genes on both X chromosomes, it would usually be fatal. For example, hemophilia or Duchenne's muscular dystrophy. So this is the example of X-linked recessive, okay? Now, the thing to remember, guys, is males more commonly express the condition than females, okay? So if the mother is a carrier, the question they might ask you is, if the mother is a carrier and the father is normal, what will the children be? So basically, 50% of the sons will be affected, but none of the daughter will be affected, okay? So let's do that again. Unaffected father, X, Y, but carrier mother. She's got the diseased alleles on one of her chromosomes. So the son will take the Y from the dad and will take, let's say, a normal from the mum. Okay, so that son, sorry, we'll take the Y from the dad and we'll take the diseased allele from the mum. So that son will be affected. He cannot mask the diseased allele with his Y. In this case, however, this son will take the Y from dad and will take the normal gene from mum, so will be unaffected. The daughter will take the X from dad, unaffected, and the X from mum, unaffected, and the other daughter will take the disease from mum, the normal from dad, so will be a carrier. So the probability here is that none of the daughters will be affected. Okay, one will be a carrier. Okay, so 50% of the daughters are carriers. None of the daughters are affected. Okay, but if the mother is a carrier, basically 50% of the sons will be affected. Okay, so that's slightly more complicated, but you need to learn that one too. The next thing they might say to you is, if the father has the condition, okay, and the mother is normal, what happens here? So in this case, the father's affected. He's got a mutation on his X. He cannot hide it with his Y. So he's got diseased X, normal Y. Mum's got normal X in both cases. So in this case, one of the daughters will be a carrier because they will, both the daughters will be carriers because they will take the X from their father. They have no choice, okay? But the sons will always get the X from the mother, so they'll be unaffected. So in this case, if the father has the condition and the mother is normal, all the daughters, 100% are carriers, but 100% of the sons are unaffected. The third scenario they might say to you is, what happens if the father has the condition and the mother is a carrier? So in that case, 50% of the daughters will have the actual condition itself. That's an important thing to know, okay? 50% of the daughters will be carriers and 50% will have the condition. And that's because, unfortunately for the daughters, basically 50% will get an X from the dad, which is disease, and an X from their mum, which is disease, so will be affected. Whereas 50% will get the X from the dad, which is affected, and a normal X from their mum, but will be carriers, okay? So 50% of the daughters are carriers, 50% of the daughters have the condition, whereas 50% of the sons will be sick. They'll get the diseased X from the mother and the Y from the father, okay? Or 50% are healthy, okay? So all of the 50s to remember for that one. So those are the three scenarios they might give you for X-linked recessive, okay? And that would be something like hemophilia or Dussen's muscular dystrophy. The last one to remember is X-linked dominant. And if you've done the MCQ part of the exam, you'll probably remember hypophosphatemic rickets. This is the one that usually comes up in the exam. So the inheritance pattern you get here depends on whether your mum or dad has the gene. So for example, what happens if your mother has it, but your father is normal? So if your mother has it, okay, remember, she's got a diseased X and a normal X, but because it's dominant, she cannot mask it. So in this case, 50% of her sons will have it, 50% of the daughters will have it because they've taken the diseased X from her. Whereas 50% of the sons and daughters will be healthy because they've taken their normal X from the dad and the normal X from the mum, okay? Females are more commonly affected than males in this one. And what happens if the father has X-linked dominant and the mum is normal? In this case, if the father has it, unfortunately, all of the daughters will be affected and none of the sons. And that's because the sons will get the Y from the dad and the normal X from the mum, but the daughters have to get an X from their dad. So they will always get the diseased one. And because it's dominant, they cannot mask it, okay? So X-linked dominant affects all of the girls, but none of the boys. Now, the next thing to say is, well, we've talked about the genotype. How do we put it together? But how does that genotype actually manifest itself as a phenotype? So how do they present clinically, okay? And two other terms that you need to know for the exam are penetrance and expressivity, okay? So penetrance is the probability that the phenotype will be expressed if you have a particular genotype, okay? And you've probably heard of a term called incomplete penetrance, and that's where you have inheritance of a mutant gene, but without the phenotype being expressed, okay? The severity of phenotype expression can also vary between individuals who have the same genotype, which is known as variable expressivity. And we see that in Marfan's, okay? So they may ask you in the exam is, tell me the difference between incomplete penetrance and variable expressivity. And the important point to note, guys, is Incomplete penetrance is where you don't express the phenotype, so you will not have symptoms. Whereas variable expressivity is where you do express the phenotype, but to varying degrees, so you will have some symptoms in variable expressivity. So we've talked about single gene, autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive. We've talked about how it's expressed, incomplete penetrance, variable expressivity, etc. So the next thing to say is, well, what about those gene defects which affect multiple genes and also other factors, for example, environmental, which can affect the way in which a phenotype is expressed? And that's basically called polygenic inheritance. And an example of that might be developmental dysplasia of the hip, or, for example, a talipes equinovarus or clubfoot. And the important point to note is that the risk of a condition being inherited in subsequent generations will decrease with polygenic inheritance. So as you go through, your risk will go down. And that's an important point, because if they ask you in the exam, to counsel the parents about a particular condition. So that's what I was just saying there. DDH, clubfoot, those are the common quoted examples. And we used to think previously that these were because of packaging disorders, but newer research has shown that they're actually caused by polygenic inheritance. Okay, guys, so I'm just going to share my second presentation. So that was genetics. So we'll just move on to embryology, and very briefly, I'll touch on fisus as well. Now, some of this will be repetition because I've talked about the limb bud before, but I think this is an important thing to note about for the exam, so I don't mind repeating these things. So when I talk about embryology, guys, for the exam, please keep it as simple as you possibly can. They are looking at clinical application. They're not looking for lots of minutiae. So it's just good to have a reasonable breadth of knowledge, okay? And I break it up into two main parts: embryology of the limb bud and embryology of the spine, okay? So let's start with embryology of the limb bud. And the important point to note is Rishi's rule of threes. This is the easiest way to remember that. 3 zones, 3 genes, and 3 directions. Okay, so the limb bud normally develops around 4 to 6 weeks of gestation. And we have 3 zones where growth occurs: the apical ectodermal ridge at the tip, the progress zone just behind it, and the zone of polarizing activity. We then have 3 directions: proximal to distal, radial to ulnar or posterior to anterior, and dorsal to ventral. Okay, so I'll go through these in a bit more detail. So first of all, proximal to distal growth is governed by the homeobox gene or HOX gene. Okay, and what happens is cells will start in the apical ectodermal ridge and then send signals to cells in the progress zone, which will then divide. And the longer you spend in the progress zone, the more divisions you undergo. So for example, in the upper limb, an arm will spend, or a humerus will spend very little time in the progress zone, whereas a phalanx will spend more time, so it will divide more regularly. So that's proximal to distal growth, homeobox gene. The next thing is the zone of polarizing activity, posterior to anterior growth, and that's under the Sonic Hedgehog gene. So if you probably all remember video games by Sega, remember Sonic Hedgehog gene. And we can see conditions where that goes wrong. So radial club hand or thumb hypoplasia I've talked about before, or for example, you might see it in fibula hemimelia. So anything where growth is affected on one part, anterior or posterior, is because of the Sonic Hedgehog gene. And that's typically with the zone of polarizing activity. Finally, growth occurs from dorsal to ventral and is under the influence of the WNT gene or wingless type gene. Okay, and an example of that would be people who have nail pads not on the dorsum of the finger, but on the volar aspect, where that may go wrong. So remember my rule of threes: 3 zones, 3 genes, and 3 directions. And as I mentioned, the limb bud forms at about 4 to 6 weeks of gestation, but something else happens about, I should have said actually 4 to 8 weeks rather than 4 to 6 weeks. Something else happens, and that's development of your other areas, such as cardiac, renal, your gut, etc. And so you commonly have associations. If the limb bud goes wrong, other things can go wrong. And you can remember that by the mnemonic VACTERL: vertebral anomalies, anal atresia, cardiac abnormalities, tracheoesophageal problems, renal, and also limb bud abnormalities. And the important point to note, guys, is if you're met with an embryological problem in the exam, they won't just give you it on its own. They'll give you it in a clinical context. And remember, it's not the limb bud deformity which might kill the patient. It's the associations. So what they are looking for you to pass the station is exclude associations such as VACTERL, VARTA, Holtorum, thrombocytopenia absent radius, and Fanconi's anemia. These are the things that you need to exclude in the exam. So that's the way that I tackle any embryological problem I meet in the exam. I describe the deformity that I see in front of me. I remember the rule of threes, 3 zones, 3 genes, and 3 directions, and I try and exclude any associated problems. So we now move on to embryology of the spine, and this is quite important to note, okay? So basically, the first thing to understand is, when does development of the spine and spinal cord start? So it starts about the 3rd week of gestation. And basically, the important point to note is this thing called gastrulation. And I'll come on to that in a second. So there's 3 germ layers which are responsible for embryological growth everywhere. And that's the ectoderm, the mesoderm, and the endoderm. So in the 3rd week of a fetus, the major event that happens is this thing called gastrulation. And that's where these 3 layers, ectoderm, mesoderm, and endoderm, form and start to become different tissues. So you start with the zygote. That basically divides to form 8 cells, which keeps on dividing until you get this hollow ball of cells called a blastula. And then it sort of seals over to form a blastocele. And then gastrulation occurs at sort of week 3. And 3 main layers form. The ectoderm, which is the outer layer, and that basically forms the nervous system and the epidermis, the skin. The mesoderm, the middle layer, forms the connective tissue. So for example, your muscles, your cartilage, your peritoneal linings, your pleura. And the endoderm, the inner layer, forms the organs and linings of the digestive and respiratory tracts, okay? Remember, guys, the neural tube and neural crest are ectoderm, and the somites and notochord are mesoderm in origin. So the next thing we move on to, and I think this is probably the most important from an orthopedic point of view, is how do the vertebrae and discs develop? And the tissue that these guys come from is the mesoderm, the middle layer. And that really gives rise to all of the body's connective tissues. And 2 buzzwords that I want you guys to remember for the exam are somites and notochords. Okay, so the notochord will form first, the somites form next. Okay, so the important point to note, guys, is 2 midline structures develop in the mesoderm: the precaudal plate and the notochordal process, which starts as a hollow tube of mesoderm, which develops from the cauda mesoderm and then becomes a solid structure called the notochord. And the notochord will then induce formation of your vertebral bodies via producing the sonic hedgehog gene that we spoke about before, and will become the nucleus pulposus. So it will stimulate, it will produce sonic hedgehog, and it then becomes the nucleus pulposus. Okay, so remember somites, remember notochord. I remember after development of the notochord, 3 structures will then form in the mesoderm. So notochord forms first. It releases sonic hedgehog gene, it becomes the nucleus pulposus, and then 3 structures form. The paraxial mesoderm, or the presomitic mesoderm, which basically lies either side of the neural tube and will develop into somites. The intermediate mesoderm, which gives rise to your genitourinary system, and the lateral plate mesoderm, which splits into a ventral layer at the front, forming the mesothelial covering of the viscera, and a dorsal layer at the back, which gives rise to your skin dermis and parietal lining of the body wall, okay? So remember, the somatic system is parietal, dorsal. The splanchnic system is visceral or ventral. Somites also, guys, they also give rise to many connective tissues. So for example, dermis, under the form of dermatome, skeletal muscle, myotome, cartilage, which comes from sclerotome, and also endothelial cells and tendons. The axial skeleton, the vertebrae, and the annulus fibrosus are derived from the somates, the sclerotome part, and the nucleus pulposus comes from the notochord, okay? So remember, axial skeleton, vertebrae, and annulus fibrosus come from the somites, that's the sclerotome part, and the nucleus pulposus comes from the notochord. Okay, and that's just showing you guys what I spoke about, those three layers, okay? So you've got the paraxial, intermediate, and lateral plate mesoderm, okay? And the lateral plate mesoderm splits into a ventral layer, which basically is your splanchnic system, and a dorsal layer, which is your somatic, okay? The paraxial mesoderm basically lies either side of the neural tube, which you can see here, and that gives rise basically to your sclerotome, myotome, and dermatome. So that's going to be your skin, it's going to be your muscle, etc., your vertebrae. And then you've basically got your intermediate mesoderm, which gives rise to your kidney and your gonads, your genitourinary system, and then your midline mesoderm, which gives rise to the precaudal plate and the notochord. Remember what I said, notochord releases sonic hedgehog gene to stimulate all of the other things to form, and then forms your nucleus pulposus, okay? So it's a little bit confusing, but if you just try and visualize that thing, okay, it's much easier to remember. So the next thing is, we've talked about the sort of structures forming around the neural tube, but what happens with the neural tube itself? How does that develop? Well, it develops from the ectoderm, okay? And it's by a process called neurulation, okay? And what happens here is the notochord will send signals to start the process of neurulation. So the signal will come from the mesodermal structure to start, okay? You'll have formation of the neural plate, a thickened area of cells, which will form at the cranial end of the embryo, and will grow from the cranial to caudal direction, okay? So the neural plate will form first at the cranial end of the embryo and grow from a cranial to caudal direction, okay? And the cranial end of the neural plate, the head, will become the future brain, and the caudal part, the tail, will become the future spinal cord. Now, by the end of week 3, you then get these neural folds. And what happens here is the lateral parts of the neural plate become elevated, and they sort of move together to form these folds, which you can see here, converging together, okay? So that's the important point to remember there. You have in the center a neural groove, which is basically a depressed region created by this folding process, okay? And then at the end of week 4, the neural folds will fuse, they'll fold over and fuse, and the neural plate will be converted into a neural tube. So you can see that here. The folding over has occurred, it's been converted into a tube, and then basically the fusion of the tube, it'll start in the middle, and then basically it'll advance up and down, cranially and caudally. And that tube will form the central nervous system, brain and spinal cord. The neural crest, basically, so during closure of the neural tube, cells on the neural crest will detach, and essentially these cells will migrate to form the neural crest, which contributes to the formation of the peripheral nervous system. So if you remember, neural tube is central nervous system, neural crest is peripheral nervous system. The ectoderm will then fold around the mesoderm and basically the endoderm. And what happens is the endoderm will become smaller and the mesoderm becomes larger. And this ectoderm will end up becoming the skin on the outside. The mesoderm will become sort of your bones, muscles, and in the middle, and the notochord becomes the nucleus pulposus. And then the endoderm on the inside will form the lining of your airways and your GI tract, okay? So ectoderm forms skin, mesoderm forms all your sort of bones, muscles, nucleus pulposus, and endoderm on the inside forms your lining of your GI tract and your airways. So guys, that's when embryology goes right. But what happens when it goes wrong, okay? So when it goes wrong, we think of spina bifida, and that's like a series of, I think of it as a spectrum. Spina bifida occulta is the most simple, meningocele, and then myelomeningocele is the worst type. And that's where the neural tube fails to close. So you have a defect and often have a layer of skin overlying exposed neural tube. And it's signified spina bifida occulta by a hairy tuft, but obviously if the neural tube itself opens up, you can get exposed dura just being covered by skin. So it can be very serious or even life-threatening. So to summarize, guys, spine embryology, remember gastrulation occurs in the 3rd week, and that's where you form the 3 key layers, okay? Endoderm, mesoderm, ectoderm. Now remember, it's the somites and notochord form, and that's the mesoderm, and then neurulation is where the neural tube forms from the ectoderm. So that's how I would break up spine embryology in the exam. I'm going to very briefly just touch on fisus to finish, okay? So in the exam, guys, you will need to be able to draw a fisus, and you'll need to know conditions which affect each layer. That's typically the way they will ask it, especially in the part 1 or multiple choice component of the exam. So in terms of drawing a fisus, I break it up into different layers. These flat layers at the surface called the germinal zone. What happens then is cells will increase in number, known as the proliferative zone. You then move into the hypertrophic zone, where cells sequentially get larger and larger and larger until they burst and release their calcium contents, which is so-called enchondral ossification. You then have your sort of metaphysis and these tortuous vascular loops, okay? And then finally, you'll have your epiphysis, okay? So those are the different layers that you need to know in the fisus. And the question you might ask is, well, what happens in each layer? So the germinal or resting zone, it's kind of a storage zone for proteins, lipids, and also glycogen. And disorders of this zone are basically any storage disorder, things like Gaucher's disease, but also pseudoachondroplasia, Nest syndrome, and diastrophic dysplasia. They can all occur in the germinal zones. Those are the ones to remember for the exam. The proliferative zone is where you increase in number. So anything which affects that can be, for example, gigantism, getting too big, or achondroplasia, too small. The hypertrophic zone is where you increase in size as you go deeper until you eventually burst and release calcium, okay? And that's typically the site of physeal fractures, and that's because you've got bigger cells and less matrix, so it's a weaker area. It's also where you get the non-renal type of SUFI as well, can occur here. And it's broken up into 3 main parts: the zone of maturation, then deeper, the zone of degeneration, and then deeper, zone of provisional calcification. The metaphysis, guys, is basically where you get the primary spongiosa, and that's where the non-renal type of SUFI can occur. And then the secondary spongiosa is where you get osteomyelitis. And I'll come on to that in a second. So remember what I said about the hypertrophic zone. 3 parts: zone of maturation, zone of degeneration, and zone of provisional calcification. And it's where the matrix is prepared for and you're initiating calcification. The metaphysis, as I mentioned, is broken up into primary and secondary spongiosa. And the reason I said is primary spongiosa is basically where you get your non-renal type of SUFI, okay? Your renal type of SUFI. And secondary is where you get your, sorry, I've lost the word. You get osteomyelitis. The reason that happens, the reason I've drawn a roller coaster is not for fun, but because if you look at the vascular loops or the way that the vessels are set up in the secondary spongiosa, they're quite tortuous loops, basically. They're like what we call hairpin loops, where it looks like a hairpin. And so what that means is blood flow is relatively slow or sluggish through these areas because you've got these hairpins. So it creates areas of stasis. So it's quite easy for bacteria to be deposited out of solution. And that's why it's a really common site, guys, for osteomyelitis. So the other question they may ask you in the exam is, and we've kind of sort of touched on that one, why does osteomyelitis occur in the metaphysis? Well, it's because you have these tortuous vascular loops, these hairpin loops. So you get relatively sluggish blood flow, stasis, and bacteria can be deposited out of solution. The next question is, why is a Salter-Harris 3 worse than a Salter-Harris 2? And the easiest way for me to do this is just draw it for you. So I'm just going to annotate this diagram. Apologies about the poor drawing. But essentially, let's say I get a fracture coming through in this zone here, a physeal fracture. Now, in a Salter-Harris 2, that fracture will exit downwards through the metaphysis, okay? It doesn't touch the progress zone or the germinal zone, sorry. So as a result, the precursors are left alone. It's got a better prognosis. If it's a Salter-Harris 3, it goes up into the epiphysis and it takes out the germinal zone. So it's got a worse prognosis. The next question is, what are the complications of physeal injuries? Well, the key thing is really, if you get a physeal arrest, it depends where it is. If you get a total physeal arrest, you're going to get shortening. If you get a partial physeal arrest, you're going to get an angular deformity. And the way that you treat this is you look at where the arrest is, how old the child is, how long they've got to go to skeletal maturity, and how big the size of the arrest is. And we call this a physeal bar. So we typically will assess this on MRI to see if it's fibrous, or on CT to see if it's bony. And we look at the size of the bar. So if it's less than 50% of the physis, then we may think about just basically taking out the bar and doing some sort of fat interposition, okay? If it's more than 50%, then we're probably going to leave it till skeletal maturity, or we may complete the arrest at a age-specific time, or we may do a contralateral epiphysiodesis, or if it's an angular deformity, we let it form and then we deal with the deformity later with an osteotomy. So these are all the ways that I think physis will be asked in the exam. So guys, thank you very much for listening. Please, if you need to get in touch with me, email me letstalkdrishi@gmail.com, or one word, or check me out on Facebook, Twitter, and I'm very happy to be asked questions, guys. Please, I love to keep in touch with you guys and help you with your progress as well. I know this is not an easy topic to get your head around, but it's something that you don't need to know in a lot of detail. It's all about learning it, but keeping it very clinical in nature. So I hope I've helped you a little bit with that, but please, if there's any further questions, very, very happy to take them.
Thank you, Rishi, for that brilliant talk as usual. And I particularly feel great about your talk is that it's purely catered to the FRCS, and everyone who listens to your talk have always found that they've really had an edge when they've written the exam. Just a couple of questions from our side. Rishi, so just to make use or to get maximum benefit from your lecture,
sure.
Can you see in a short time you're going to be an examiner as well? Can you say the top 3 questions from the first 2 talks? For example, the embryology of the limb bud and genetics. What are the questions, and how would you answer those questions?
So the key thing, guys, is
if you want, you can put the slide as well, because
let me do that. It's the easiest thing to do. So remember, they are testing your ability as a day 1 orthopedic consultant. They are not testing your ability as a geneticist. That's the important point to note. Sorry, I'm just going to reopen that. Right. So what they want you to do is they want you to keep it very clinical. And the way that they'll ask you the questions, again, is going to be very clinical. So the thing that I would learn here is all of this stuff here. It's not that important, to be quite honest with you. I was just telling you about mitosis and meiosis. This is more biology, A levels and things like that. This is what they're going to ask you. They're going to show you clinical scenarios, and they may say, what has happened here? So they may show you, typically in the UK, they show Turner's, exo, they show trisomy 21 Downs, because this is something that we often meet in clinical practice. And you have to say, as an orthopedic surgeon, what do you want to know about Down syndrome? Well, you want to know what are the orthopedic manifestations of it, what are the non-orthopedic manifestations of it, and how can I help as an orthopedic surgeon? So the management is multidisciplinary. It involves geneticists, orthopedic surgeons, pediatricians, and you may need to appropriately counsel the parents as well. So that's all I would really learn about that.
Anything else on Downs? Clinodactyly, is it? The small little finger?
Correct, yes. Clinodactyly as well, yeah. And then also the other thing as well I should have said is with Turner's syndrome as well. They typically have the cubitus valgus deformity as well. They can get webbing of the neck and also be of short stature as well. Okay, so those are the three abnormalities that I would probably have a quick look at. Klinefelters, Downs, and Turners. Yeah, and just understand that those are whole chromosome abnormalities. Okay, structural, again, I wouldn't get too hung up about this. What they will ask you about is modes of inheritance. This is the way I've seen it asked in the exam. So they will typically show you a condition. They'll never start with, tell me about genetics. They will always start with the clinical case. So they may show you something like an autosomal dominant condition. They may show you an achondroplasia patient, because that's a patient we often meet in the exam. They may say, what are some of the orthopedic problems that an achondroplastic may have? So short stature, they may have spinal problems, etc. And then they may take you on to, if you were counseling this patient about the chances of them passing it on to their children, what would you say? That's a typical question they will ask. So what I say to all my trainees is practice drawing out the Punnett square, which is what I was showing before. So what I mean by that, for each of those 4 conditions, practice drawing this. If the father is affected, if the mother is normal, what percentage of children will be affected? I've pretty much put every question they've asked in the exam before. So I don't mind just leaving that slide up just for a second so people can have a look at this. I've just taken this picture off the internet, guys. So it's not something I've drawn, but these are the typical scenarios that I've seen given in the exam. So if the father's affected, the mother normal, what percentage of children will be affected? Because this is typically what you want to counsel the parents about. Next, autosomal recessive. If both parents are carriers, so they have the diseased allele, what percentage will have the disease and what percentage of carriers? Again, you're being asked to draw it. Okay, and I'll leave that up there for a second. X-linked recessive. So this would typically be your Duchenne's muscular dystrophy, your hemophilia patients. If the mother is a carrier and the father is normal, what happens to children? So 50% of the sons affected, 0% of the daughters affected. Now, I have to say, I'm sure many of your students are far cleverer than I am. And I was one of these people that I couldn't just remember facts off by heart. I could never retain the information. So for me, rather than learning these percentages, you will never retain it. The easiest thing for me is just practice the drawings. And I really encourage you, all the students watching, please practice. You know, when you're watching TV, you know, whatever you're watching, just this is something you can do on the train, you can do on, you know, if you're driving in the car and you're a passenger, whatever, just practice it all the time. So you become very, very regular and seasoned at it. The next scenario, if the father has the condition and the mother is normal, what happens to the children? And then finally, if the father has the condition and the mother is a carrier, what happens to the children? So all of these Punnett squares, this is the main way that I've seen genetics asked in the exam, Hitesh. They seem to really like these, and I've seen them ask candidates to draw these out. What happens if the mother has it and the father is normal? That's for X-linked dominant. So this will be your hypophosphatemic rickets. Okay. And finally, what happens if the father has it and the mum is normal? Yeah. And the other thing I was going to say, the other tip is, even if you can remember these percentages off by heart, you'll actually lose marks if you just come out with the percentage. They actually want to see you draw this, because what they want to think is, okay, this candidate, he or she is able to work this out. They don't want to say, oh, he's just memorized it or she's memorized it. So even if you know the percentage in your head, still draw out the diagram or the Punnett square. Okay. And you can make it simple. You can draw a stick man, a stick woman. It doesn't have to be anything complex, okay? Again, penetrance and expressivity, I've just mentioned very quickly. But for me, the other reason I've seen things asked is polygenic inheritance as well. They've sometimes shown developmental dysplasia of the hip or clubfoot, and they've said, what causes this? And most people used to believe these were packaging disorders, but now we think it is mainly polygenic inheritance. So that's sort of genetics, where I think that will go. Then in terms of the last two, embryology and fisus, where do I think this will go? Again, they will show you a clinical scenario. So they will normally show you something like fibula hemimelia or radial club hand or hypoplasia of the thumb. And they may talk about management of that. And I've done a previous talk on radial club hand, so you can find that on Orthopaedic Principles channel. But they may say to you what's happening at an embryological level. And then you can bring in your 3 zones, 3 genes, 3 directions, and you should be able to draw the limb bud. Okay. And again, in terms of spinal embryology, they'll probably show you somebody with spina bifida. Okay. And they may say, what's happened here? So I would first describe the normal embryology of the spine, and then I would say the step where the closure happens. And I'll just show you one here. So this is the step where the neural plate fuses to form the neural tube. That's the bit that goes wrong, guys, in spina bifida. That's the bit that fails to close in week 4. Okay, so that's where you bring this into your answer. All right, and then finally, the way that I've seen fisus asked in the exam is I've seen them again show a condition and say, why is this happening? And that's your perfect opportunity to draw the fisus. They love it when you draw diagrams in the exam because it shows understanding. Okay, so they may ask you, where does Salter-Harris 2 fractures occur? Where does Salter-Harris 3 fractures occur? Where does achondroplasia occur? Where does this occur? Why are children more susceptible to, osteomyelitis than adults? And it's because they've got this very tortuous blood vessels. So bacteria gets deposited out of solution. Why is a Salter-Harris 2 better than the Salter-Harris 3? What are the complications of fiscal injury? So that's typically how they will ask it in the exam. It's always to do with the clinical first.
Thank you, Rishi. I think that's all the questions that we have for this session. Thank you for yet another fantastic lecture. And I'm sure this is going to benefit a lot of people all over the world.
Thank you so much for having me, guys. And it's lovely to speak to you once again.
Bye-bye.